It's my pleasure to introduce you today the LBRN, the Distinguished Biomedical Research Seminar Series invited speaker, Dr. Russel Middaugh from the University of Kansas at Lawrence. Dr. Middaugh got his bachelor's degree at the UC Santa Cruz, and then he got his PhD degree from Cornell University in 1978. He then did some postdoctoral work in Minnesota and got his first faculty appointment at University of Wyoming, where he was for about 10 years. Before he moved to Merck to develop the gene therapy and protein formulation group over there. And then he decided to go back to academia, so he went to the University of Kansas at Lawrence, where he's been since about 15 years, and is the Takeru and Ava Iguchi Distinguished Professor of Pharmaceutical Chemistry. Is really a biophysical chemist. He has appointments in chemistry and physics, a large group of people, about 30 people working in his group. And so it's my pleasure to be here today to hear about all the his gene therapy project and the protein formulation and vaccine development. So the title of his talk changed just a little bit. Is phase diagrams and macromolecular stability. Very interdisciplinary again. And thank you for coming.
Thank you. Thank you. Yeah, don't worry about the title. This is an old title, and I didn't bother to change it. And I would be hard put actually to provide an accurate title for today's talk. So you'll see why as we get into it. So as you were just told, my background is very different than most people because I spent almost 10 years in industry. And that 10 years in industry was devoted primarily to characterizing various macromolecular systems, everything from peptides and proteins to virus particles to gene delivery agents. And that was very focused ultimately on developing products. So many of you may know products that I developed. So my group at Merck developed the vaccines for human papillomavirus, for HPV, for hepatitis A, for the shingles vaccine, for rotavirus, and for many of the combination vaccines that Merck has developed. And that has very much influenced my research interests in academia, some of which are basic, but I have to tell you that most of our support actually comes from either industry, or our biggest supporters are charitable organizations like the Gates Foundation and the Sabin Institute, who we work with on the development of vaccines. But in the process of doing that, we do some fairly fundamental work, and I'll touch upon that today. The talk will be somewhat diverse in that regard. Now, if you want to develop a protein as a drug, or a virus, or a peptide as a vaccine, you're faced with the problem of maintaining its structural integrity. And I'm going to skip a lot of slides, and rather than rearranging my talk each time, I just add things and then skip slides. In the old days, the way we did this is by measuring everything we could possibly measure. And we still do that, but what I'm going to tell you today is we do it very, very differently. And so typically, this is actually, I was actually, while at Kansas, the director of vaccine formulation at Biogen for many years. I couldn't find someone to be head, with good reason, I think. And so I did that, and I tried to automate their whole process of the characterization of their proteins. And this was successful with a number of different proteins that they now have on the market. And so there's a whole flurry of methods. I won't go through them because I'm going to touch upon many of them later. And what you do is you tend to take your molecule, stress it with things like temperature, pH, ionic strength. You might shake it. You might freeze it and thaw it a number of times, and then see what happens to it, and then try to minimize those degradative reactions. They can be non-covalent, and I'll focus on non-covalent reactions today, but they can also be covalent. And so various chemical changes like deamidation, oxidation, and the like all occur as well. And you have to deal with those issues as well. I'm not going to talk about those today. what I'm going to do is to tell you about a new way of analyzing proteins and related macromolecular systems. And believe it or not, we've actually applied this to something as complex as a bacterial cell. It's been used quite successfully with viral particles and viruses themselves. So you can actually apply this technique to very complex molecular systems. But I'm going to illustrate it with a really simple system. And this is the way we actually originally developed the mathematical approach that we use. None of it is particularly complicated, as you'll see. It uses really pretty much all established technology. It's just the application is somewhat different. So here are some ultraviolet absorption spectra of proteins. And I guess all those things flying around there, I'm not doing that, am I? I hope. Okay, I guess that's okay. All right. Anyway, so most of you, if you've worked with proteins at all, you've seen these type of spectra. They're pretty nondescript. Here's these are just standard model proteins: serum albumin, RNA, lysozyme, CON A, and they're characterized by this maxima right here. Usually, it's a little bit above 280 nanometers. And if you look carefully, there's usually this shoulder, and as I'll show in a minute, the shoulder comes from tryptophan residues. And then there's often some structure. If you look carefully, you'll notice that there's some undulations here. You see them better with serum albumin, and that's reflecting the vibrational structure of the phenyl side chain, the phenylalanine, right? So for a long time, it was thought that such spectra were useful only, for example, measuring the concentration, if you do the extinction coefficient. I'm going to show you can get a whole lot of information and use this technique that we've developed. All right. So here's the second derivative of the 3 aromatic amino acids. And you can see they're in very characteristic regions. And here's a protein in which we've taken the second derivative. So based on these model spectra right here, you can assign these peaks. So here are the phenylalanine peaks. Here's the tyrosine peak here. There's a tryptophan and tyrosine peak very close. This is a combined peak. And then the far peak is a tryptophan peak. Here's the effect of temperature on those peaks that you just saw. Now, as you can see, the effect is very regular. There are no surprising deflections here. It's an intrinsic effect of temperature on the ultraviolet spectrum for tryptophan, tyrosine, and phenylalanine. It might surprise you to know that there are error bars. Usually, if I look hard, I can find one. You can actually, there's one right there. They're actually error bars, but the points are bigger than the error bars. So you might wonder, well, how does one achieve? Notice this is only these are only 0.2 of a nanometer. These increments right here. How is it possible to get such precise measurements? Well, all of the work I'm going to describe is obtained either using a diode array spectrometer or using a spectrophotometer with a CCD detector. And so this allows you to determine the absorbance at any particular wavelength with very, very high precision, better than 1 to 200ths of a nanometer. And we take advantage of that in the work that I'm just about to show you. But it's important to note this regular behavior. Now, I'm not going to talk about it, but actually the slopes of these lines, we have developed technology using these slopes to measure dynamic aspects of protein structure, because this reflects the dynamic behavior of the side chains and its interaction with the solvent. But I'm not going to talk about that. We have a series of papers in protein science about that if you're interested, okay? The technique we're going to use, though, to analyze this type of data is something we call an empirical phase diagram. So here's a description, but I'll give you a somewhat different verbal description to try to make it even simpler than it is. What we're going to do is we're going to represent our molecule of interest, our molecular system of interest, as a vector in a fairly highly dimensioned space. The components of the vector are simply going to be the experimental measurements that we make. The example I'm going to start with is those 6 peaks that I showed you, and each peak will be a component of a vector. So we'll represent a protein as a vector with 6 components, which corresponds to each of those peaks. And I'll show you we can also do this with many techniques and combine that all together into an even more comprehensive picture. What we then do is we perform a procedure most of you will probably be familiar with called singular value decomposition, to extract from that the 3 components of the vector that make the biggest contribution to the final multidimensional vector. And then we'll use an RGB, red, green, blue color scheme to assign colors to those 3 most components, combine them into a color. All right, so here's an illustration of this. Here's data. We won't be concerned yet exactly what the data is. And here's a result of this in which at each, and this is temperature on this axis and pH on this axis. Each of these little bars right here, data are obtained here at 1 pH intervals and at about 2.5 degree intervals. So each of these little bars right here is a vector produced by the 3 major experimental components, which by the way are normalized, so they can be compared to one another. Now, so what this shows you is states, not thermodynamic states, I want to emphasize that, no equilibrium is applied here, but structural states of the system, and with a state defined by some sort of uniform color. Transitions in states often defined by gradual changes in this color. In the case of highly variable states, such as aggregation, multicolors, etc., you're going to see lots of these diagrams today, and also I'll show you different versions of these diagrams which don't use color. But so what you got here is kind of, in this case, a temperature pH stress response picture of a protein. And so it, of course, doesn't have the information that a 3-dimensional structure in terms of the coordinates of the atoms has, but it has a lot more information in it than the use of a single technique, because not only are we using a lot of techniques or a lot of pieces of data from a single technique, but we're also using stress, temperature, pH, ionic strength, whatever we choose, to increase the amount of information. Okay, all right, and this is just a mathematical process to do this. It's very simple. Let me just illustrate, though. Here we've got our 6-component vector. This is a second derivative spectrum. Here are the positions of the peak. Here's the vector. So at any one temperature and pH, you've got a vector, okay? And so here's the results of this from the red, green, blue color scheme as I just showed you. All right, okay. Now, we've just recently, this is unpublished work, began to develop different versions of this. And so you're the first audience, actually, to hear about this. So one criticism, or at least one question we've always been asked about the previous type of diagram was, what do the colors mean? And the answer is that the colors didn't mean anything. They were completely arbitrary. They were just selected by the singular value decomposition process. So what we have done recently is we've tried to make the colors mean something, okay? So up above, what we have are some, this is what, CD, circular dichroism, intrinsic fluorescence, peak position, and static light scattering. Just pick 3 different kinds of measurements in this example. I'll come back to the absorption example in a minute. And what we do is we normalize this data, and we produce what we call indices. In the case of CD, secondary structure, tertiary structure. Everybody follow me why I say that? And finally, the aggregation index right here. So they're just normalized values from 0 to 1. And now we're assigning the meaning based on alterations in this vector will be alterations in secondary structure. Alterations in this, alterations in tertiary structure, at least as sensed by changes in the environment of tryptophan residues in proteins. Although you see we have many other ways of doing this. And then aggregation, we measure some version of light scattering. Okay, now this is another example of the same thing, just showing you what the data looks like. That the previous one, was serum albumin. This is one of the many proteins we work on. This is a protein that's in development for developing a staphylococcal vaccine from a colleague. And the second, so that's one type of phase diagram. We'll call it the index type phase diagram that's different than the conventional phase diagram I just showed you. But we developed these new ones recently, and one of the reasons is my colleague, David Volkin, I guess he won't be embarrassed if I say this, he was colorblind, and I'm 65, and so he's going to be my replacement for head of our large group. And we realized that we rely so heavily on these phase diagrams, you'll see why, okay? And he couldn't see the differences in color, so that was not such a good thing. And so he said, well, can you do anything else? And I thought about it a little bit, and here are the 2 solutions we've come up with. You can decide whether they're of value, whether they're just humorous, or people who have seen them have reacted quite differently to them. All right. All right. Now here's the index method right here, in which we assign colors. Now, if the native state is, we use red and green for the native state. When you add them together for the secondary and tertiary structure, you get yellow, etc. Molten globule states. Everybody knows what molten globule states are. You know, states where the tertiary structure is disrupted, the secondary structure is still intact. Usually these states tend to aggregate. They're really important to pharmaceutical chemists because aggregation is the big bugaboo of developing vaccines and therapeutic protein products, okay, et cetera. And here's a more expansive view. Here's a typical empirical phase diagram. Here's the same phase diagram based on these indices. And I put over here, here's the behavior of the tertiary structure, secondary structure, tertiary structure, and aggregation index. And these are added together to make this. So you can actually now make statements about what's happening in these different phases just by looking at the color. A small improvement, but I guess in importance, people seem to want it very badly. All right? So the other two methods we've developed are based on something called a radar chart, and the other one on something called a Chernoff face diagram. So in the radar chart, what we're going to do now is not use color, but we're going to represent the normalized value of our method by a position along a polar axis. So if, let's say we use a method here, method 1, and it'll have a value from 0, which is the center here, to 1. And so for any temperature and pH, just choosing those as the ones, the methods I'll talk about, the stresses I'll talk about, you can plot a value somewhere along these axes. And then for each method, you can plot a value. It could be 0, it could be 1. But those values, of course, will range depending on the temperature and pH. So each temperature pH compound will generate one of these diagrams. What we're going to do is we're going to connect the various points along this to create a sort of distorted polygon. You'll see these in just a moment. So now the values are represented simply by the length of from the center to the vertex of this. And so any protein will now become, its behavior will be described by this polygon. Okay? And then we can do the same thing here, except we use facial features. So we can change the mouth, smiling or unsmiling. We can add a mustache if we want. We can change the angle of the eyebrows. So as many different features as you want to put, you can have methods that will go into the diagram. All right, what's it look like? Well, it's a bit bewildering when you see one of these at first. So here are the polygons here. You see it's just a point when nothing's changed. So everything's relative to the native state. But then you see these this is only using 3 methods now, so only 3 vertices. Sometimes we use as many as a dozen. It gets a little complicated to look at. And here are the faces right here. I don't think you can see them too well, but I'll show you bigger versions of them, okay? All right, let's get that. So, okay, now I'll come back and show you more of these diagrams later. But now let me show you the simple example. Here's a protein we worked on very early on. We originally developed the phase diagram technique using this protein. It's kind of a funny story. This is a protein from Pfizer, and it actually came from the Pfizer veterinary division. And they called us many years ago and said, well, we're developing this as an alternative to bovine somatotropin, and also it has antibiotic activity as well. And so the idea was that you'd give it to cattle, and you wouldn't have to give the cattle, for example, antibiotics. There wouldn't be any antibiotics in the milk, and everybody would be happy. Of course, as soon as they showed this to me, I said, this is never going to be a product. You'll never be able to produce this cheaply enough. I mean, no cattle rancher is going to pay $20 for an injection or something. It's never going to happen. It's too expensive. And they did eventually drop the project. But one of the advantages of their dropping that project was they had tons of the protein. And they called me once and said, before we dump it out, do you want any? And I said, sure, send us lots of it. We'll use it as one of our model proteins. So that's where our protein came from here. All right. So I'm going to skip through slides very rapidly because the values of the individual data here are not very important, all right? But I want you to get a feeling for the data, for the patterning of the data, all right? So in this figure and in the next 6 figures, all right, what we have is the values of the emission, of the absorption maxima of the 6 peaks in the absorption spectrum, all right? So you can see that for this peak, which is a phenylalanine peak, not much happens. But with this peak, another phenylalanine peak, you'd see a transition, see a big transition here, okay? See, for the tyrosine residues, you see a transition in the opposite direction. A little transition for that mixed peak, and then a big, nice, pretty transition for the tryptophan residues. Okay, now, if you think about it for a minute, in proteins, the phenylalanine residues are almost all buried deep in the interior because they're so apolar. So they're a probe of the protein's interior. most tyrosine residues are interfacial because of their hydroxyl group, and they tend to interact with the water. Tryptophan can be anywhere. It can be very buried on the surface in any number of locations. So each of these peaks is a probe of a different kind of environment. So the fact that they don't change in the same way is not surprising. But what that means is this is a very information-rich picture of what's actually happening to the protein. Now this is pH 2, all right? Now watch what happens. Just allow your eyes to relax. I don't want to spend a lot of time talking about it, but watch how these spectra change. pH 3, pH 4, look at that. That changed direction. Okay, all of a sudden there's a peak down here, okay? pH 5. Now notice the noise is starting to appear. The protein's starting to aggregate. It's hard to measure peak position when it begins to aggregate. pH 6, pH 7. Okay? All right, it's a pretty complex collection of data, but okay, we could summarize it, couldn't we, with a chart and just say, well, here are the different peaks and here are the different pHs and here's the initial position of the peak. Here's where the transitions start. Here's where they finish. If you can measure them, here's the midpoint of the transition, et cetera, et cetera. All right? It's not intuitively very useful information, but we can summarize it in the form of a phase diagram, an empirical phase diagram. Empirical, I maybe should emphasize that point, because we want to differentiate this from a true equilibrium phase diagram, which at times it could be, but mostly it's not. All right? So here's the first one we ever did, and this looks quite a bit different than most of the ones I'll show you, because in these days, we were using Mathematica to do the mathematical analysis, and we switched over to MATLAB later. And so it looks somewhat different, but the data is the same. All right, so you can see here's this dark green region at the higher pH. There's this intermediate green region, then there's this other region. Remember all that aggregation? That shows up as this multicolor region. And these different high temperature unfolded states are differentiated as well. All right. All right, now, we could do the same thing, but we aren't limited to using a single technique, the 6 techniques of ultraviolet absorption. All right, so here's an example where this is an example at pH 2, in which we've used a bunch of different things. We've used circular dichroism. Here's differential scanning calorimetry. There's FTIR to look at the amide 1 band, et cetera, a whole bunch of different techniques. Today, it doesn't matter exactly what those techniques are. Each of them monitors the protein in a somewhat different way, although their information may well be overlapping. Okay, and here's pH 3, 4, 5. See everything just jumping around like crazy. 6, 7. Okay, and here's a little chart summarizing the data. Not very useful. In contrast, this is using the MATLAB method. Here's the phase diagram. You'll notice it's very similar to the phase diagram that we obtained from the simple measurement of the ultraviolet spectrum. But you can see there's this big area, which is the native state, and you can see this transition region, the aggregation region, these high temperature, low pH states, which of course are highly unfolded. Okay. Here's the same thing done at higher ionic strength. Massive aggregation. You can see how different it is at high ionic strength. All right, now I'm going to go through a series of examples, and I'll try to tell you a little story with each example, so you at least know what the kinds of things. These are examples of things that we've been interested in over the years, and show you phase diagrams. I won't spend a lot of time talking about the individual data, okay? Unless somebody sees something and has a particular interest. So here's a project that we've been working on for a long time, and that's the development of a vaccine for ricin. So I think most of you probably know ricin is a protein that's found in the beans of the castor plant. It's about the easiest protein to isolate you can imagine. Unfortunately, it's one of the most toxic substances known. It's been used as a weapon by terrorists in the famous subway attacks in Japan, for example. This was the agent that was used. It's been used by I guess, the famous example, if you ever go to London, you can go to the Terrorism Museum in London. You can see the umbrella that they seized from a Bulgarian Secret Service agent who assassinated someone on the streets of London with the sharpened point of an umbrella, simply flicked it and had ricin on the tip when they walked by. And the person just felt a little something and scratched and didn't think anything about it and dropped dead a day later. So needless to say, there's an interest in developing a vaccine, probably first for the military, but later, if terrorists were to start to use ricin more extensively, a vaccine would presumably be something that would be very important to have. So there are a couple of places working on this. So the project that we work on is one that was developed actually, and the protein has 2 mutations in it, so it's not toxic anymore. It was developed by Ellen Vitetta over in Texas. She licensed that to a company, which is now, it was called Door Biopharma, it's now called Sologenics. And we've been working with them. We have, believe it or not, multiple grants from the military and from the NIH trying to develop this vaccine. There are a lot of problems because the vaccine's not very immunogenic and it's not very stable. All right, I won't say much more about it because what I really want to talk about today is representing the protein. All right, so here's that second derivative spectrum of ricin, okay? And here it is, here's the results at 5 different pHs. And here's a phase diagram. And I've just shown a limited region of the phase diagram right here. This is just 10 to 60 degrees and pH 4 to 8. To show, but what you'll notice is that up here, that there's some melting of the protein already at 50, actually by 40 degrees, you're starting to see it. So it's not very stable, all right? So now what I'm going to show you is just a series of measurements of the ricin molecule at different pH values. And here's simply the light scattering. In this case, all we did was measure the turbidity, the optical density at 360 nanometers. And you can see that even in the most stable form, by 35 degrees, you're beginning to get some aggregation. It's very unstable, although it's very pH dependent. Right? And then the whole collection of data. This is, I'll go through it quickly, fluorescence intensity from the tryptophan residues, the peak positions in the fluorescence experiment. It starts to aggregate out here. That's why it drops that way. Okay. Here's a circular dichroism spectrum at 10 degrees. You can see there isn't much difference, but of course when you start heating it, of course you see significant differences in stability. Not surprisingly, the less stable forms are at high pH 4 and at low pH at 4 and at high pH at 8. And the rest are pretty not as temperature dependent, all right? Now, we frequently probe structures using fluorescent dyes. With proteins, we tend to use either things that detect apolar regions like ANS, 8-enolenonaphthylene sulfonic acid, or we use things that detect intermolecular beta structure. There are a number of dyes that do that. When we're looking at DNA, there are a whole collection of dyes that bind into the major and minor grooves and that intercalate. We use membrane-active dyes when we're looking at viruses that have membranes, all of which probe some aspect of structure. And we typically probe as a function of things like temperature, pH, although other things as well. Right? Here's ANS in this particular case. ANS tends to be very weakly fluorescent water, but when it hides away from water, when it binds to a protein, the fluorescence goes up. And of course, that's what you're seeing here, the binding of the dye. All right, and this is just the peak position in the fluorescence experiments. And here's a phase diagram now made from all of those measurements that I just described, an empirical phase diagram. So if you remember back, it looks very much like the phase diagram from the second derivative method, but of course it's much more information rich. And got this graduated area in here, and these correspond to the molten globule states. And the reason you see this is because, of course, you see changes in tertiary structure before you see changes in secondary structure. So the tertiary structure starts to change here. And it gradually, and then when the secondary structure changes, you get another change in color right here. And in this region, you have these states, which, as also shown in the phase diagram, highly aggregate. Okay, another example. This is recombinant protective antigen, which is the protein used in anthrax vaccines. And you all know about anthrax. There's more money to study anthrax than anything else because the terrorist attack in the U.S. was against, of course, our senators and representatives, and so they took that very personally and have supplied and continue to supply large amounts of money to study this sort of thing, if any of you are interested. All right, we've worked on versions of vaccines here from, I'd say, about 10 different companies and organizations, including the military as well. And this data is from a collaboration we had with Becton Dickinson, which was interested in novel delivery systems. They wanted to deliver this as an aerosol, the vaccine as an aerosol, probably the same way it would be delivered as a bioweapon as well. All right, but I'm just going to show you solution data. And once again, it's a collection. I know you can't read the axes down here, okay? All right, this is just aggregation. So this is pH, and this is temperature. These are different pH values rather right here. This is just time as a function of the stress. Okay, and this CD, and so here's all the data. I won't go through it. Here's a phase diagram. This was one of the original ones. It still has these funny little things in here from Mathematica. You might wonder, well, how can I write these things in here? This was before we actually made the colors mean something. But what you can do, of course, is go back and look at the actual data. And you can say, well, is the circular dichroism changing? You can measure, if you like, the amount of secondary structure at the temperature and pH that you have, and then make statements about what's actually happening. But the important thing here, from a pharmaceutical point of view, is these boundaries. So we call them apparent phase boundaries, because they're the places where things start to change. And when we're trying to develop something like a vaccine or a therapeutic protein, you need to have it be able to stand storage on a shelf somewhere at, say, 4 degrees in a refrigerator. Could be minus 80 if somebody had a minus 80. Most people don't, okay, out in the real world. But you need to be able to have your molecule as stable as you possibly can. Otherwise, it simply won't function. One of the reasons we've had trouble delivering vaccines to the developing world is simply the fact that we can't get the vaccines to where they need to go. You don't think about that sort of thing, but a box full of measles vaccines, it's a live attenuated virus, very sensitive to temperature, very easily can be left out on a loading dock in Nairobi, and in 10 minutes it's dead. It's not an active vaccine anymore. In fact, when people have gone and looked out in villages where vaccines have been delivered, often they find that they're inactive. And the ugly secret is that I will tell you that we knew at Merck, because the same thing's true in the US. It shouldn't be, but it is. If you go into your doctor's office and check the measles vaccine, sometimes it won't be active and it'll be much less effective. You can still produce an immune response, but a much weaker immune response. The trick is to make them more stable so you don't have to worry about that. Anyway, here's an example of something that isn't a vaccine yet. We've been working on this for years. Respiratory syncytial virus causes upper respiratory infections, most commonly in children. Many of the upper respiratory infections your kids get are from this virus. There is a therapeutic protein that's used to treat serious infections of this type, made by MedImmune. Okay? But obviously, that's not a desirable thing. By the way, it's $20,000 a dose, I think. It has to be given IV. And so having a vaccine to prevent this would be very helpful. Unfortunately, what happened was many years ago, when they tried to make an attenuated live virus vaccine, they caused more disease than preventing it. And there's a long story about why that happened I won't go into here. But so the decision was made to drop the program for many years. Wife, now Pfizer, picked it up. We worked with them for years on this. They finally decided not to go forward. The program now is at MedImmune, who sells the actual other product. So I sometimes wonder if this moves slowly there because they're going to build a vaccine, of course, then they won't need their other expensive product anymore, which sells over $1 billion a year, by the way. These infections can be quite serious. Anyway, so this is a virus. It's a live attenuated virus. And you can make the kinds of measurements we just described with something like a virus. There's a big difference, though, isn't there? An obvious difference. Whereas in a protein, we can make some statements about, hey, this is the change in secondary structure of this protein. Here, a viral particle will have multiple proteins. It'll have some nucleic acid. And so you're going to be measuring, when you measure a signal, you're measuring a signal that's a composite of the behavior of all the components in their response to temperature, pH, whatever you're using. Now, you might think that would prohibit the use of this, but it doesn't. Because you can still make the measurements, you can still see changes, and it often turns out that the inactivating events that you're concerned with are still reflected in these measurements. They may not be quite as large, but they're still there, and you can still use attenuation of those changes to develop more stable forms. And this works, and as I say, it worked with rotavirus, which probably all of your kids get a rotavirus. You know, we used to kill 2 million kids a year with rotavirus infections, and now in the US, there are almost none. It's almost wiped out by the vaccine. It's oral, by the way, just squirt it. Had to stabilize it to stomach pH, just put it in a really strong buffer that way. And it's starting to work now out in the developing world as we add subtypes. There are 4 different types of viruses in the rotavirus vaccine. We're starting to add some of the rare subtypes that are more important in other parts of the world. So And there are many others as well. Measles, mumps, rubella, varicella, those are all live attenuated viral vaccines. They have to be stabilized, and that's the tricky part of them. Okay, here's the second derivative spectrum. So you actually have some components from the nucleic acids in addition to the protein components. Okay, here's shifts as a function of temperature at various pH values. Okay, and this is just a summary of the optical density data. It tends to aggregate between pH 3 and 6, but it doesn't aggregate up here. It's a little bit of turbidity here because the virus is big particles, so it scatters a significant amount of light. Okay, here's more optical density. Here's fluorescence intensity, and here's relative circular dichroism measurements. You can see how broad the transitions are, and that's because you're seeing a sum of transitions in this case. Right? I won't go through all the data. Here's a phase diagram, though. All right? Now, hopefully, I'll have time to come back and show you how this phase diagram was used to develop a formulation. This thing's not very stable, okay, as you can see. Right? But we were able to dramatically stabilize it by picking conditions from this and doing high-throughput screening of small compounds and finding stabilizers. And if I have time, I'll get to that and show that to you. Here's a virus-like particle. This is one we continue to work on. This is work with a company called Ligasite, which is located in Bozeman, Montana. Nice place to have a company. I'm on their scientific board, and I always enjoy my trips there. It is a virus-like particle. Now, what is that? There are two other virus-like particle vaccines, hepatitis B and the human papillomavirus vaccine. And so what you do is you just isolate the major surface protein, and it self-assembles into a particle that looks like the virus. Probably doesn't come as a big surprise to you, that the information to produce the viral capsid actually resides in the three-dimensional structure, and it will spontaneously assemble. If you take away the nucleic acid, of course, it's completely non-infectious, but still highly immunogenic. And so that was done here. This norovirus is a virus that is a subclass of something called noroviruses. There are a lot of different types of noroviruses. Norwalk is the virus that causes shipboard illnesses, and sometimes in hotels they get these outbreaks. This is the virus that causes that. Interestingly, we thought for years that that was going to be its major application. So the company that developed a small company and was looking for some small income to survive off of this. But then something wonderful for the company and sort of shocking happened. And that was that as the rotavirus vaccine became more widely used, it was discovered that half of the respiratory infections between 0 and 2, usually a child would get 2 infections, almost everyone would if you aren't vaccinated. And it's a diarrheal disease. And if you dehydrate too much, dead, you got to get into the hospital fast to have one of these infections. Everybody should have this vaccine, all kids, so there should be no problem anymore. But when they looked at the incidence, it dropped in half and stopped. But I just said 100% were protected, right? Well, the reason is because it turns out this is the cause of massive respiratory infection. Nobody knew that. It wasn't recognized. So this now is potentially a really important vaccine. It's been in the clinic. It works. It's been in the clinic in challenge models. You go to a university, and I remember we did this with our DNA vaccine at Merck with flu. And what you'd really like to do is a challenge study. So challenge studies are pretty shaky. It can be a bit unethical. That means immunizing someone with a vaccine you don't know that it'll work, and then challenging them with the organism, seeing if they get sick. Well, flu, pick healthy volunteers, and you can get FDA approval to do that. Of course, anyone in such a trial has to understand the danger, and the trick is to offer a couple $1,000s to participate in the trial. So where do you go find a big, healthy audience that for $2,000 would do just about anything? Yeah, universities, right? Merck did Johns Hopkins. You'd think that would be a place, and they had to turn kids away, right? And that's how this was done as well. And it was challenged, and it works. It actually protected like 70, not 100%, because there are multiple different types of norovirus. But the 2 that were included in the vaccine, it did protect 70%. They're trying to increase it now. There's still some arguments about whether we need this vaccine, but we won't go into that. I think we do, but then I'm prejudiced. So Anyway, so here's data. Okay, this is just a picture of a virus-like particle right here. There's an actual three-dimensional structure from cryo-EM. This is the protein that's used to create the virus-like particle. So here's the CD at 10 degrees. Okay, and you can see it doesn't change too much. Maybe it's something subtle at pH 8 right here, but that's about it. Okay, and here's the CD as a function of temperature. All right, this is just plotted in terms of secondary structure change. All right. Here's that second derivative ultraviolet analysis. We picked only 3 peaks in this case for the work, and here's some phenylalanine data right here, and here's data from fluorescence tryptophan emission as a function of pH. And I won't go on. It goes on. We actually looked at the particles. We actually used the images as one of the components. You can see at low pH, the particles are starting to fall apart. You can actually visually see that. Okay? I'll skip the rest. Here's the phase diagram. This is what I wanted to show you. Here are the different regions. You can assign the regions by going back and looking at the actual data. Now, what we would do is use that index type phase diagram. That was not done with this. Right? And I'll skip this. This is just on some interesting mechanistic information. So I have a bunch of examples. All right, not very interesting, I guess. So I'll go through them. This is a kind of a color slideshow. All right, everybody ready? All right, here we go. Okay, we've worked on botulinum toxin. So maybe I should tell a story about that. We formulated Botox at the University of Kansas. People are always shocked to hear that. This is the most toxic substance known. Everybody probably knows, right? A few nanograms, yeah, you're okay. And that's why people use it cosmetically. A microgram, oh, you've got hours and you're dead. All right? It's really toxic. So how do you study something like this? Well, we have a biological safety level 3 facility that we use. Sometimes we work on the AIDS virus and things like that, so we want to have a protected facility. So this was brought into that facility. I also have had a physician, he's left now on my staff, who had the monoclonal antibody that you can give as an antidote to this. Okay? But the key was getting us the protein, because to make the kinds of measurements I just described to you, you can't measure nanograms of protein. You need many micrograms, 10, 20, 30 micrograms of protein. So for us to do our work, we needed probably 50 milligrams each time we would perform a large series of experiments. At that time, the total production of Botox by the company that made it was 10 milligrams a year. Okay, that's enough, by the way. That's how potent it is. They make one lot, and then they collect their $500 million. Not a bad deal, huh? But we needed lots more. And so they ended up bringing it to us by special jet with a military escort. And then a bunch of police cars would go out with cops who were armed, and they'd actually bring it to our laboratories. I mean, it's special security in the laboratory when we worked on it. It was very strange. And I think that I wouldn't do it again. We were lucky. We didn't have any problems, but we were able to pull it off. Anyway, here's a phase diagram from that work. Interestingly enough, the botulinum toxin, when it's used as a drug, by the way, it's a legitimate drug. Most of you know that, right? It's used to reduce spasms in the eye, for example, and other types of. It's a fantastic drug, by the way. You know, there are people who get eye spasms. They're terrible. And you inject this drug, and bang, that's it. And it works for like 6 months. And they don't have any problems. So besides its cosmetic use, it has real, legitimate medical uses. But the toxin that's given actually has complex to it carrier proteins. And so here's the actual phase diagram of the neurotoxin. And you can't see this very well, but what we did was we took these associated proteins and the neurotoxin and then the toxin itself, which is the combination of the two, and did difference phase diagrams in order to look at and ask the question whether there were structural changes when the complex formed. And the answer was yes, and I won't go into the interpretation of the data right now. We've done a lot of work on adenoviruses. Adenoviruses are one of the most popular viruses for use as gene delivery agents. However, once upon a time, there was a vaccine for adenoviruses. There was a massive outbreak of adenoviral infections in military bases, and Louisiana was one of the places, Fort Polk, where there were these massive. I did my basic training at Fort Polk. You know, I just remembered that. Right this second, I've been here before. Fort Polk, Louisiana, which without a doubt was one of the most miserable places I've ever been in my life. And You know, and I remember out on March, waking up with a coral snake in my sleeping bag. It didn't bite me, fortunately. I totally forgot. I wiped it out of my mind. Right? Yeah. Anyway, the military actually, through Wyeth, developed a vaccine for this. And then the FDA said now the military vaccines have to be subject to the same rules that civilian vaccines, and that killed the vaccine because it was kind of a slop-together vaccine, and Wyeth didn't really make much money off of it, so they discontinued it. Now they're having massive outbreaks of adenovirus infections again, so I think we're going to see the vaccine come back, probably a better quality vaccine. But we worked on it as a gene delivery agent because you can completely strip an adenovirus of all its genes and put anything you want inside. It's a little bit tricky, it's by co-infection of cells in culture, but you can do that, and they efficiently infect cells, and as a consequence, you can deliver genes in this way, and that's why we were working on. Here's a phase diagram. Oops, wrong thing. Adenovirus type 2. We've done non-viral delivery agents on which you complex DNA, and this is an example with a cationic lipid. And here are phase diagrams, although notice in this case we're using ionic strength and pH. So this is ionic strength and pH. Temperature, the DNA is pretty stable. It doesn't start to melt till pretty high temperature. So I have lots of these. I'm going to skip by them. These are for different types of charge things at different pH. Okay. Now there's another type of phase diagram that we've been making lately, and what it does is it includes in it information about the dynamic structure of a molecule. So everything we made, all the measurements that we typically use are time-averaged measurements. Now, so what we did in this case was we added measurements that were sensitive to the internal motions of the molecule. So what am I talking about? Well, isotope exchange. We use acoustic spectroscopy. We use something called pressure perturbation, differential scanning. Calorimetry, we use red edge shift measurements, a variety of others as well. And we did that. What we found was this is an IgG. It's a monoclonal IgG. Here's the static phase diagram for this molecule. We found this region right in here. And if you look at the data, you find that at fairly moderate pH and at lower temperatures, there's this change in the dynamic structure of the molecule that occurs. And what we've been working on lately is the effect of stabilizers and destabilizers on this region to see if we can develop compounds that take advantage of our ability to measure this region and improve the stability of molecules. And that would be a seminar unto itself. So anyway, this is the very first phase diagram we ever showed of that. Okay, and here are just a bunch of examples of the new types of phase diagrams that we're talking about. So these are these indices right here, and here's oops, let me go back. So here's the index phase diagram, and here's a version using radar charts, a version using Chernoff faces. So what we've done here is instead of showing all the individual images, we set a statistical limit on how much an image or a radar chart could vary, and then we just defined the region that contained those images and presented it as that image to simplify the diagrams. And you can see native state, very happy, okay? Unfolded state, unhappy. Massive aggregation, look at the hair growing up there, right? Very unhappy, all right? I don't know what you think about this. I kind of like it myself, but, you know, I don't know. We haven't submitted it for publication yet. I think we'll send it to Protein Science, and we're either going to get one or two reactions, either, well, this is interesting, and they'll publish it, or they're going to write and say, are you out of your mind? Don't ever send us anything like this again. But we'll just have to see how they react to that. Anyway, so I have a bunch of, this is for serum albumin, but here's the data. Here's one for, this is one of those staphylococcal proteins. This is the hemagglutinin, the major protein in flu vaccines right here. You can see how complex these diagrams are, okay? Aldolase, a model protein, chymotrypsin, okay? This is another, skip that. Now, another use of phase diagrams, different from this, is to look at mutants. And I picked one example here. This is a paper that just appeared in Protein Science, in which we compared different mutants of fibroblast growth factor 1. Now, this is one of those proteins you get attached to, and I started working on this at Merck, and I have all kinds of stories about this protein. But we still work on it even today, because there's still a company interested in it. I won't say it failed in the clinic, because it worked in the clinic for Merck, but they decided it wasn't worth developing. It basically enhanced the growth of diabetic foot ulcers. You rub it on in a cream that we developed, and it would speed up that healing. But it turned out there was a big placebo effect, that people don't take care of their foot ulcers, and all these people in the clinical study started taking care of them. So 30% the placebo worked, and about 60% with the growth factor worked. I thought it was enough to develop the protein, but Merck thought it wasn't, and I didn't go forward with it. But anyway, with colleagues, we've been, Mike Blaber at Florida State, in particular in this case, we've been trying to make the protein independent of the binding of polyanions. Now, this is a tremendously unstable protein. At room temperature, it aggregates and unfolds almost immediately. And even at 4 degrees, it doesn't last longer than a few days. But it turns out that the protein binds to polyanions. And it does that in its natural environment. It binds to the proteoglycans on the surface of cells. And apparently, the molecules will line up and they'll cause receptor aggregation, tyrosine kinase receptors, and they cross-phosphorylation, the whole thing. But it's very intimately involved with these proteoglycans. So when we formulated it with a simple polyanion, heparin. And the heparin stabilized it so you can sit out at room temperature for 2 years. That's how dramatic the effect is. But you have to have heparin, and nobody wanted heparin in their formulations, especially in a diabetic foot ulcer. So over the years, we've been working on mutating the protein, trying to separate the receptor binding capability and its ability to activate its various signal transduction pathways from the polyanion binding. We're making mutants, and we found such mutants, suffice it to say, and I just wanted to show you here's some phase diagrams of these mutants, and you can see, here's the wild type right here, but you can see how much we can push things up, for example, with some of these mutants. And phase diagrams show this very, 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 very nicely. And so one of the problems with the technique, how are we doing time-wise? Not too good, okay. So one of the concerns in the industry and among colleagues has always been, well, does this mean that I have to have a circular dichroism spectrometer, a fluorometer, a DSC, an FTIR system, et cetera, et cetera, et cetera? And the answer was yes, okay? But we wondered if there might not be a way to make enough measurements in a single instrument. And we thought that because of the ultraviolet absorption measurements, which worked pretty well. They weren't as good as the multiple methods, but suggested that if we could get like CD, fluorescence, light scattering in one instrument. And so we've been able to do that in collaboration with 3 companies. And so I apologize, and I'm not going to say which I like more, okay? I don't want to advertise, but the original work was with Applied Photophysics and their Chiroscan circular dichroism instrument, in which you can also get, it's a very sensitive instrument, so you can scan from the far ultraviolet, which gets you secondary structure information, through the near ultraviolet in a single scan. It's the only instrument I know of that will do that. And that gives you tertiary structural information. But you can also get light scattering information by looking out at the turbidity at 350. And they also have another detector so you can do fluorescence as well. And so here are phase diagrams created, just examples of phase diagrams from this particular instrument. It's reasonably compact, it's kind of pretty too, and works real well. Instrument's expensive. A complete setup to do all this would be well over $100,000, but that's the way she goes. Another instrument that does the same thing, we talked to a company in Georgia called OLIS, Online's Instrument System, and we outlined in block diagram form, because I knew they built separate instruments, and said, well, here's what we want. We want a CD machine, which they made already. We want a fluorometer. They made one of those. We want a light scattering system. We want we want a diode array absorption system. Oh, by the way, the Chiroscan uses a CCD detector, so you can get high-resolution second derivative detector. Here, it's done through a diode array. And they made this thing. It fills it's kind of big. It fills a complete bench. It's got a 6-position cell holder in it. The Chiroscan has a 4-position, but the Chiroscan has an auto sampler, so you can still do a lot of samples. Okay, and here are phase diagrams created from what, if you could read it right here, what they call the protein machine. They actually named it after me, the Middaugh protein machine. You can buy one of these if you want one. We have the only one. Nobody's bought one yet. I don't know why. I don't know why. But they're going to offer this software. And of course, you can make all the different types of phase diagrams as well. And then lately, we've been working with this. It's a microtiter plate fluorometer. Unfortunately, the microtiter plates can only be used once, and they cost $300 each. And we're working with them trying to change that. But what this instrument does, it will measure fluorescence emission spectrum, has a separate dye, a separate laser that you can use for light scattering. So you can measure multiple fluorescence signals plus light scattering and make phase diagrams out of that data. And this is the highest throughput of the three instruments. And I don't know if you noticed or not, this is all the same protein, and all three phase diagrams look the same. It works perfectly well. And furthermore, we've compared them to single instrument phase diagrams. They all work well as well. All right. Now, what I was going to talk about, maybe I better stop. If people say, if I hear yelling, don't stop, please don't stop, then I won't stop. But what I was going to talk about was how you really use the phase diagrams now to do high throughput screening. And I was going to show that using the conditions from the phase diagrams, you can pick a set of conditions, screen, develop an assay, screen libraries of compounds of various sorts. I was going to show you a library, a little simple library that we invented using small synthetic compounds in which you always find stabilizers, always, 100% of the time. And then you can optimize the stabilizers for concentration. You can use them in combination, put them in and create a formulation of a product. And that formulation then can be used for the actual drug or vaccine. But because we're running late, I will stop right there and just acknowledge. Oops, hold on here and just go to my acknowledgement slide. You can see the results of. Oh, aren't you sorry you missed that slide? Okay, okay. But, we'll skip all this. And this by the way shows remember that I said how stable the RSV was? Well, we found that polyanions, just like FGF, stabilize RSV, and that was the key. Everything on this slide, everything on these slides is published except for the initial description of the Chernoff phase diagrams and the radar diagrams. Okay, and I'll skip that and just acknowledge most of these are people who've long since left the laboratory. Let's see. Oops, that's another talk. Lisa's at the NIH. Latoya's a professor at the University of Colorado. Hai Hong is at GSK. Fernando's at Novartis up in Canada. Laura is at a diagnostics company. Sangeeta's still with us. She's our lab manager. John Ralston is the physicist who wrote the software for the original development of the phase diagram. And we do that ourselves now. But I'll tell you a story of John Ralston. So I got the idea to do this, and I knew something about quantum mechanics, and so I thought, well, we'll make big matrices, et cetera, et cetera. But I wasn't quite sure what to do exactly. So I asked one of my friends, and he said, oh, go talk to this theoretical physicist. He's interested in everything, and he'll be able to help you. John Ralston in our physics department. And so I went over, and I explained everything to him. And he said, this is easy. You know, I know exactly how to do it. I'll just send me the data, and I'll create this empirical phase diagram that you want, and which he did in a couple of weeks. But as I was leaving, and I was halfway out there, he said, Russ, he said, I have one last question. And I said, what's that? And he said, what exactly are these proteins you're talking about? And I realized he had, like physicists want to do, he had seen the problem totally in terms of this abstract entity. It didn't really matter what it was. He saw the solution without even knowing what it was. And I have one of his students in my lab. He's doing his PhD with me now. And maybe I'll mention that one of the things that they've been working on, and it actually is completed now, is one of the things we'd like to be able to do is to compare proteins and see the differences. In the pharmaceutical industry, as patency labs, what's happened is that people are starting to make what are called follow-on biologics. In other words, they're not truly, well, they're when you make a protein, and one company makes a protein, another, they're not exactly the same. Right? There can be subtle differences. So, but if we're going to have people make proteins, so it'd be cheaper, that's the idea, right? How can we make sure that they'll be cheaper? Well, one way is to say they don't have to do a clinical study with 40,000 people, which costs $100 million, right? And so the idea is, well, if you compare two things structurally and say they were the same, even though you might have to check it with a few people, you don't have to do these big, massive studies. And so we've been developing methods using phase diagrams to allow us to compare things. But we decided to do it in a very different way, in which what we do is we use, in any one element of the matrix, is the entire spectrum, the entire CD spectrum, the entire light scattering spectrum, the entire fluorescence spectrum, the entire differential scanning calorimetry thermogram. whatever it might be. And so at any particular temperature and pH, we'll have this whole spectral set now. And then we use a color system, a functional color system, and then we compare the two and do a statistical analysis and print difference diagrams that show color where they're different. And that's worked extremely well. And we've tested it with very subtle mutants and things like that, and it seems quite sensitive. And that's we're just thinking about where to publish that. Probably as a note somewhere. Anyway, and I won't go through everybody else in various locations, but we are always, as everyone else is, of course, in debt. Much of this work is done as a service to companies. And so they own the intellectual property. We surrender it to them. The only thing we retain is the right to publish it. And we'll give them a month or two if they want to write patents or whatever. Every once in a while, they forget to do that, and I end up on their patents, and that's not a good thing for them. Not a good thing at all. I'll sign it over, but it makes the trouble. Anyway, I'd be happy to answer any questions here or elsewhere.
Any questions?
Surely someone, and you're welcome to ask very critical questions. I've certainly been asked them before, especially in industry, about this work. Should I expect a mean question then? Huh?
Yes, exactly. I always have a question. Yeah, so why not proton NMR?
Well, okay, why not proton NMR? And proton NMR would be ideal in many ways. The problem is the size of many of the molecules. And if we really want good information, we need the label, right? And, well, I'm saying detailed information, okay, about distances between atoms and things like that, which is what you really would like. And for obvious reasons, that's not useful to us because that's a different molecule, right? So with proton NMR, just measuring the peaks and their positions, we've made phase diagrams from proton NMR spectra. And it works reasonably well, but the you know how much they overlap in various regions of the spectra. Even with the best deconvolution methods we could use, the phase diagrams that came out of that were no better, and in many cases not as good as using these other techniques. So that's the reason. I also don't have an NMR in my lab, although I do have access to a 700 and an 800. So, but you're absolutely right, though. And we tried the same thing with FTIR, which has multiple peaks. So the amide 1 region has 7 or 8 peaks that you can deconvolute. They're all sensitive to secondary structure. The side chains in FTIR are almost all so, their vibrational spectra is so weak you can't see them, right? So that's a problem. The technique we have not done it with, where I think it might work, is Raman. Right? Now, Raman's very insensitive. You need very high concentrations of proteins, at least several milligrams per mil. By the way, most of this work can be done at 1/10 of a milligram per mil or lower. And so that means companies will sometimes come to us with a milligram of protein. We can make a phase diagram and reproduce it with a milligram of protein. And that's very important early on in development. Later on, it's not so important. It's a good idea. No one else? You're hurting my feelings by not asking questions. Yeah, okay.
Okay, I'll ask another.
Okay.
So when we do have nitrogen-15 labeled proteins and we're looking to see that they are structured, a lot of times what we have to do is we have to go through iterations of trying different buffers, different salts, different concentrations. So what you're doing is like that, but do you go into the detail of trying sodium versus potassium?
Oh, absolutely, absolutely. The slides I flipped through very rapidly showed exactly that sort of procedure. You know, there was a really interesting, I wonder if you know this paper, an NMR paper quite a few years ago, not before you were born, but in which an NMR group was concerned with stabilizing proteins, and they hit upon a magic combination of 2 compounds, and it was, I think it was aspartic acid or aspartic or glutamic acid and lysine or something like that at a 1-to-1 ratio. And we've tried to confirm that And we've been unable to do so. It's always bothered me because they did like 5 or 6 proteins and it stabilized them all. And yes, we do sometimes see that those amino acids will stabilize, but there was nothing about that combination. We've looked at it 100 times. We've never seen it. So, yeah, same concentrations they used. Now, I don't know what they saw. I wish I knew what they had seen, but we didn't see it. Yeah, when we screen, we look at a library known as the GRAS library. Does anybody know what that stands for? GRAS library. Anybody want to guess? GRAS. Generally regarded as safe. Okay, so this is a collection of compounds that have been recognized through their oral use primarily as safe. And when companies come to us and they want us to try to create a formulation, they prefer that we stick to that library. It's about 50 compounds. And it's carbohydrates, polyhydroxylated compounds, some polymers, amino acids, nothing. Detergents, that's a big one, various types of detergents and amphiphiles. However, we've created our own library, which includes all kinds of interesting compounds, including polyanions, polycations, and things like rare types of osmolytes that are used and are found in various organisms, and on and on. And we frequently pull really good stabilizers out of there, and we're still waiting for companies. They would rather have a less stable compound than take a chance and try one of these. The FDA does not say they can't try them. You can try them. The thing they fear is that they'll discover during development that one of them is toxic, and then they have to create a new formulation. But we test them with cells, and they're not toxic with cells. That doesn't mean a lot. And we can inject them into mice and make sure the mice don't drop over dead. But the really extensive stability study or toxicity studies you need to do, we haven't done. And so they're reluctant to go forward with those libraries, which is those compounds that we identify in those libraries. I wish that wasn't true, because I think we could have a lot more, especially some more stable vaccines, but no luck so far. Only one person asked questions. Now, do you want me to go away from Louisiana thinking, yeah, no, I think they're all gone. Or maybe they were shut off. I don't know. Anyway, no one? All right, well, thank you very much. I appreciate the opportunity to talk to you.